and pick out specific nucleotide binding proteins. And the importance of this is that when we have disease states such as cancer, most of the oncogenes are the proteins from oncogenes are nucleotide binding proteins. And, okay, back up.
We're using this mic.
Okay. Thank you. Okay, so I can just walk wherever I want. All right. So what I would point out is basically I'm a chemist, biochemist type. I'm not an Alzheimer's research individual, and I'm not a person that really is involved much in toxicology. And so the work you're seeing, I think no one, at least they can't accuse me of having any vested interest in the outcomes of the research that we're doing. And so what I'm going to try and point out today, we're using this new technology. It's reasonably new, but it's used all over the world. These probes are used for a lot of different things, and for those of you that are interested in protein chemistry, you can look up my publication. They're very selective. They bind to specific proteins, and not only do they bind to specific proteins, but they bind to specific sites on that protein. So we can pull out the GTP binding domain of a huge protein like microtubulin. The other aspect of this is that using this, we have found different nucleotide binding proteins that are missing in different diseases, and this is published. And many years ago we published, not that many years ago, but a few years ago, that you could not have normal interaction of GTP, which is required for normal tubulin polymerization, especially in brain tubulin. That in AD brains this was dysfunctional, whereas in controlled brains and brains from other demented diseases this seemed to be normal. And trying to hunt down the cause of Alzheimer's disease, you consider a lot of facets. And through a large time of research in our laboratory, we found out that perhaps mercury might be involved. Other people have made this speculation, which we'll talk about. And so in doing this, I would want to make this statement very clear right now. I would not say that mercury causes Alzheimer's disease. That would be expanding my data beyond what I would like to say. I would say that mercury in a chelated form is extremely neurotoxic in very, very small amounts, much smaller than probably most people would consider, and that's what our data is about. So the first part of this talk, if I can get this going, is going to be just a little bit of, am I not getting that to move? I'm pushing forward. Oh. Okay, we're okay?
You got to turn it on.
At any rate, bear with me the first part of this. If you don't understand this, the photochemistry, and it was working, it was just going the wrong direction. If you don't understand the first part, you'll definitely understand the autoradiograms because they're black and white. But I feel like if there are people here that know a little chemistry, I owe them something. And this is just a problem that I started on as a graduate student. It's a problem of active site labeling. And that's if you want to pick out a specific enzyme. It has an active site with a lot of different amino acid residues in here, and you had a substrate that kind of fits into this site. What you do is you would covalently modify this, add some chemically or nucleophilic reactive group that would go sit on here, and because of the proximity might react with something in this area. As you can see that sometimes the amino acid residues, such as put here by leucine, they're not very reactive. And again, if you modified it with a sulfhydryl-directed reagent, you might have a sulfhydryl in the binding site, but you're just as likely to hit one that's outside. And this led me to the some work I read actually by Westheimer many years ago called photoaffinity labeling. And this gave me the idea of making probes of nucleotides. This is ATP, GTP, cyclic A, cyclic G. They're the energy and regulating components of the body. And what we're going to change on this is the only change, and we'll talk about the entire structure, is we're going to put an azide in this position or elsewhere on the molecule. And the unique thing about an azide is that you can explode it with light. You shine light on it and it literally explodes, giving off nitrogen gas and generating a nitrene, which is one of the most reactive chemical species known. If photoaffinity works right, if you can make the molecule bind to the right enzyme, whatever it's bound to, it will stick to at that time. It will make a covalent attachment, as we've shown here on this first reaction, which is called the insertion reaction, and the only one we need to worry about at this time. You generate this while it's bound to the protein. They're doing its job, and it will displace even a carbon-hydrogen bond if it's held close enough and in the right proximity, and assuming you have a nitrene that is reactive enough. It can also add across double bonds, do a nucleophilic attack, and it can do the abstraction too, but we have never seen this occur in our biological systems. But this is the general chemistry that's occurring with light. This is the structure of the molecule, or part of the structure we're going to talk about today. We put an azide on ATP. This is just an ATP molecule with an azide attached. And we make the phosphates either alpha or gamma radiolabeled, and this is how we tag the protein. The bottom line is we can take this molecule, we can put it into a system such as a muscle contracting system, a salt transporting system, or a tubulin polymerization system. And without light, these things will mimic the native compound. They'll cause muscle to contract, they'll hydrolyze this phosphate, transport sodium, or they'll cause tubulin polymerize. You shine light on them, and they immediately kill that system wherever they bind by generating a nitrogen through this molecule. Now, this is the three-dimensional structure of this, and this is for those interested. This is the space-filling model, and you can see that it binds and has a structure very similar to ATP, with the exception of this blue region coming right out here. That's the nitrine. When it explodes, it leaves a place that would be just this short. So this molecule is very similar to the nucleotide triphosphate in three-dimensional structure. Now, starting my career, I have an addiction to horses. So I started my career at the University of Wyoming, and that's the reason I also went to the University of Kentucky. At those places, you have to worry about your equipment. This is the expensive equipment you need to do this type of research after you make the probes. And you can do it in light, just like this, because these are activated, most of them, by UV lamps. You use a little 190 microwatt UV lamp. You have a room that's lit as well as this. You need an ice bucket, and you need a place to place your samples and mix it, and something to block the photolabel. And this is how I started out. I'm a little more elegant than this now, but this is how we started doing a lot of these experiments. And what's not shown here, though, are the hands of a good graduate student or postdoc that really wants to work hard and do something. And all the work we're talking about today was done by, excellent graduate students, and mostly that I've had at the University of Wyoming and University of Kentucky. The way we check these compounds, and to show you their photoactivity, even with a little lamp, this is a UV spectra of the compounds, and this is how fast they photolyze in seconds with that little handheld UV lamp. And this tells you that the explosion or the generation of the nitrine is going very effectively, because as the molecule blows up, it changes its absorption profile. And so using this, we can say that even if we had a very concentrated solution, we would be free through photolysing it in 70 seconds. We routinely use an absorption coefficient that would be down about where my red light is right now. And so we can get through photolysing usually in 20 or 30 seconds with a very, very gentle photolysis system, not something that would inhibit the protein. And now the concept we're going to talk about here, and this is what, if you want to critique my data as you're sitting here, if I'm telling you the truth now, and I am, and this has been verified by other people who use these probes also, I should be able to mix my probes into a crude extract of brain, in which I run that out, the gel out, we'll see, say, 200 protein bands. And if my probes are working a lot right, I will only label a very few of those bands depending upon the concentration in the probe used. So I don't label proteins just that are there, only those that bind the nucleotide. And this is shown in this cartoon where we're talking about before photolysis, we can have the molecule binding to a specific protein that has a shape that would accept it at its active site, not binding to others. And in this procedure before photolysis, we can look at the substrate properties. We can look at phosphorylation, any enzymatic activity, reduction of NAD, etc. And then when we're ready and we're convinced that we are interacting with the right molecule, and we know the KD, the KMs, and everything that's involved, we can hit it with a flash of light or a few seconds of gentle photolysis, and we'll get specific labeling of this protein, and almost none, or essentially none, of the non-specific labeling where we just in general run into it. Non-specific labeling does occur, but it's a minimum compared to this. Now, I'll get through the chemistry. The other thing that we can do with this, because this is important, people can ask the question on many disease states. The enzyme can be there, but there can be something wrong with its active site, and we're working on some problems like this now. The probe, if we talk about this being a huge molecule that would fill half this room, the probe only sets in a binding loop or a couple of binding loops at a specific location on the molecule. We have found and reported, and this is just for information, this is published in JVC Biochemistry and several other journals, that we can pick out the amino acid residues that make up this binding loop from a crude protein. So these things are very, very selective. And you'll have to take my word for that unless you would challenge.
Are those pi pieces nucleotides?
This is the nucleotide. This is just the photoprobe. We make other things besides nucleotides, but the pi piece is the photoprobe. It would represent the nucleotide with the azide. And what we're saying is that we can hit 2 or 3 of the amino acid residues, including the backbone, in the protein loop that binds the nucleotide that causes the conformational change that actually does the work. when you use the energy of a nucleotide. No. So this is just the structure of a zero-GTP. We've talked about ATP in the initial part. This is a zero-GTP. It looks very similar to ATP, with the exception it's different here and here. And that's the reason we put the azide over here, so we could make a molecule that would distinguish between ATP and GTP binding proteins. And this just leads me into the introduction. What we're going to talk about now are some findings that we found with Alzheimer's disease and published a few years ago. What this is a Kamasi Brilliant Blue stain gel, the proteins from Alzheimer's or progressive supranuclear palsy. And this is when I was young and naive about biomedical research, and I called these people normal. Well, they're not normal, they're dead. I mean, they died of something, but they didn't die of a dementing disease. So we call them controls. You have to pardon this thing. And what we've done here is we've separated proteins, and if you look through this, you don't find any changes that you can count on that are reproducible between AD and any of the other control brains, and we've looked a lot. There's very few protein changes you can see on the level of Kamasi blue staining. We mix this with a zero-GTP, which is P32 labeled. It's in every one of these lanes, and every other one, and it's just at a 5 micromolar concentration, which is relatively low, and we hit every other lane with light and every other lane without light, because if there's phosphorylation occurring, we want to see it. And after we do this, we know that some of the proteins in this level are labeled with a zero-GTP that are G-binding proteins that bind GTP very rapidly. They won't be the oncogenes, the slow-exchanging G-binding proteins, but things like tubulin and other GTP interacting. Over here to the right, this is something we did after we knew what we were talking about, is alpha and beta subunit of tubulin, the beta being the bottom, the alpha being the top. And this will be shown on the next autoradiograph, which will expose only the proteins in this system that are radio-labeled. This is the autoradiogram made from that prior gel. If you can remember, there's a crack through here. You can see it going through here also. And this is the same system. And we'll not dwell on this a lot, but you can see that with the 2 subunits of tubulin, we only label the beta, and it co-migrates with this protein right here. In every system where we photolized, every other lane where we hit it with light, we label this 45 kilodalton protein. This protein here happens to be tubulin. We found out it's a beta subunit tubulin. We'll show you how in a minute. And you can see that in Alzheimer's disease, this protein does not interact. The other AD is over here, and it doesn't interact. Quite different from the supranuclear palsy and the 2 controls, which are the individuals who have died of other diseases. The protein at the bottom that labels in every one of them makes a good internal control. This turns out to be actin and glutamine synthetase, and we'll talk about glutamine synthetase at the end of this talk, because we're using it now to diagnose Alzheimer's disease in the cerebral spinal fluid. But what we can find and can absolutely say, and we've done a screen on this, is that Alzheimer's disease, the beta tubulin does not bind GTP very well. This is a screen we did of several AD and control individuals. You can see the first 4 are controls. We've gotten a little more sophisticated now, and we photolized them, incubated them for a few hours to see what would happen with the exposure levels, and you can see it doesn't change, but every one of these labels in the beta tubulin area. Now over through here, you can see some of these have none. There's a couple of them that have some labeling. It's usually quite a bit lighter than what you would see in the control, but roughly we find that about 9 out of 10 or 8 out of 10 of the ADs won't photolabel beta tubulin at all that are Alzheimer's disease. We find maybe 2 or 1 out of 10 that will photolabel lightly. Usually it's much less in relation to the amount that's in the bottom band. We have to do a division. Now what this says is there's something dramatically wrong with tubulin in Alzheimer's disease brain. And we were setting out thinking, if this could be the cause. Now, for those of you that don't understand, tubulin is a dimer, and it makes a microtubule. It's what holds the neuronal cell in its shape. And it is also, quote, the railroad track, if you'll use a paraphrase or an analogy, of what takes certain compounds such as glutamate, neurotransmitters, and dumps them outside of the cell, maybe even the waste products. It also brings things in from the outer cell into the cytoplasm and into the nucleus. So it's the railroad track. You don't have to get rid of all the tubulin to wreck it. All you have to do is pull out a few rails. So this is a very significant find, and I'll have to admit that I'm quite surprised that people, I think because they're wed to amyloid protein, haven't considered this a more important finding in Alzheimer's disease. I think later on they probably will. Now, to prove what this protein is and how we can use these probes, this is just, again, a control brain. As we can see, the control brain labels in both the top and the bottom band. And we have a 0 GTP in here, and we're putting in a compound to protect. We're putting in ATP, and ATP is totally different than GTP. As we increase the concentration of ATP in this direction, we can see that this band disappears immediately. This is the lower band, and that's glutamine synthetase and actin. They're both specifically ATP binding proteins. So ATP goes in and binds to their active site and prevents GTP from binding there, and they prefer ATP to GTP. However, the top band, which is missing in Alzheimer's disease, isn't protected by ATP at all, indicating that it's very, very specific for GTP. The next slide will show an autorated graph where we change this. We change the ATP to GTP, and you'll see a marked difference. With GTP protecting against the photoprobe that is its mimic, we get almost total protection by the time we get out to 10 to 25 micromolar, which is exactly what we would see with purified tubulin. And you can see that the 45 kilodalton band doesn't protect very well at all, and that's because that protein prefers, it doesn't care which one it sees, GTP, ATP, etc., so it's not as nearly selective. And if we plot this data by cutting these bands out and doing counting, the data comes out like this. ATP doesn't protect very well at all. GTP protects very rapidly, and if we do a saturation, we do see a saturation indicating it's not non-specific labeling, which we weren't worried about since we're only labeling 1 or 2 proteins in that crude extract. And using this type of technology, this is how we go into the brain, AD brain and other diseased brains, or in the cerebral spinal fluid, and we can pick out proteins, tell you whether they prefer ATP, GTP, cyclic A, cyclic G, NAD, or any of the nucleotide binding proteins. And by doing a very simple set of experiments like this, we can identify them. Now, also, after finding this, this is just a Comanche blue stain gel, and what we found out in AD brain is that if we look at the AD and the supernatant, almost none of the tubulin is existing there. If we compare this to a normal control, we found out that about 40% of the tubulin is in a cytosolic fraction. So whatever is happening in AD brain, it's taking the fraction of tubulin that's not polymerized and putting it in a non-polymerizable, in a highly polymerized form that's not normal, can't interact with GTP, and it goes down into the pellet. So we have some idea of the location. So again, here's a model of the microtubules for those that haven't followed that. What we're looking at is GTP binding to a small dimer such as this. This will come around and it will form a microtubule. And this is the system that holds the neuronal cell in shape and also allows little motors to run along the outer surface and transports things in and out of the nucleus and in and out of the cell. And we did some experiments, early experiments, when we were looking at it, and this was the effect of different cations on the zero GTP labeling in AD and normal brain tissues. In this particular case, this is an AD, and depending upon the additions, we can label the bottom band and we can't label the top. But the reason I put this up was to show two things. This is a control brain, and we can change some things here by adding calcium, as we have calcium in this lane, or EDTA up to 10 millimolar, calcium 1 millimolar, and magnesium 10 millimolar. And you can see that we can affect this bottom band here, which is an ATP binding protein somewhat, but we don't affect the tubulin labeling in this control band at all, with at least 10 millimolar EDTA. This is very important for the future work, and with 1 millimolar calcium. Calcium is known to be an inhibitor of tubulin polymerization. And so we talked about Alzheimer's disease and how we would study it, and I did a lot of reading in this, and there are a lot of, there's many hypotheses about what causes Alzheimer's disease, as there are days in the year, I think. And the one that kind of attracted me to it was the environmental agents and toxins, because it didn't seem to be something that was tied in too much to absolute genetics, although there are certain forms, which most of you are pretty well aware of, that do cause this. So we started looking at possibly the top two for the most, and hoping that this would help us answer any question if something appeared down here. And to tell you how old this disease is, it was first studied, and defined in about at the 1890s up to the 1900s, and it's been around a long time. They were looking at degenerating neurites, and in 1907 and in 1963 they came up with two things that could be considered maybe the hallmarks, two of the other hallmarks of the disease, but you have to be dead to have this done unless you like having brain biopsies. And so what is really needed for Alzheimer's disease for quite some length of time is an anti-mortem test, something where you could do a biopsy or do a test to find out whether or not this would be important. And I've read a lot of literature about the paired helical fragments and how they're related to cytoskeletal proteins, which may be involved in this thing. This is another item, and I'm going to try and get through. I think I'm stuck. We're getting into the mercury right now. But you had to have the back of this to show
Could you go over what you're saying about the EDTA, what effect that
it had no effect at 10 millimolar. I just want you to remember that. I mean, that's important for the rest of it. Because the first thing you look at when you start saying tubulin doesn't photolabel in Alzheimer's disease brain, I was worried it was something I was doing, that maybe they had tubulin that was more sensitive to EDTA or some fraction like that. And what we're going to find out here, and what I'm going to try and present to you, is an argument that mercury chelated by EDTA and maybe chelated by certain other things such as glutamate is very neurotoxic and at low concentrations, much, much lower than 10 millimolar. Looks like one of my slides tried to eat up the projector, or the projector did that. Yes?
You just stated that calcium inhibits tubulin synthesis?
No, polymerization, high concentrations, yes. And that's what we thought, maybe there was an elevated calcium in the AD brain that might have... Pardon? Intracellular, yes, intracellularly. It has to get at the location. If you have tubulin in a test tube and you add GTP, it will polymerize, and you can measure that polymerization, and calcium will prevent that.
Of course, mercury initiates an increase in intracellular calcium.
Yes. Yes.
Another question. What causes the neurotoxicity of EDTA? Do you know the mechanism of that?
Yes, we're going to talk about that. It's what it does. It's the toxicity that everyone has described with mercury interacting with sulfhydryl groups. What we're going to show today is that it is the complex of mercury with EDTA that is most destructive to neuronal tissue, based on the tubulin polymerization. And with small amounts of mercury chelated to EDTA, we can take a control homogenate and make it look exactly like an AD homogenate. An Alzheimer's disease, I'm sorry. We'll show that on the slide here in just a minute, hopefully.
So it potentiates mercury somehow?
Yes.
Like it potentiates iron and free radicals?
It potentiates mercury only with regard to its interaction with tubulin. It has no effect with mercury on other enzymes that we've tested. Only with tubulin.
And permeability?
This is not my work. Other people have reported that mercury chelated with EDTA is more permeable through the gut, gets into the bloodstream faster, passes through cell membranes faster, which you would expect.
Blood-brain barrier too?
Sure. Mercury won't exist as a biochemist and a chemist. I can tell you mercury won't exist free in any biological system for more than a fraction of a millisecond, because it's just got too high affinity to all the amino acid residues that are in proteins.
What, elemental mercury or mercury?
Mercury ion. Mercury ion. Elemental mercury, of course, would be, yes.
The machine, Dr.
Yes, I didn't think my slides... Okay. Okay. Yes?
Do you think that says anything about the use of EDTA as a clinical chelating agent?
I sure do. I would not recommend EDTA. Well, we'll tell the story. If you look at the structure of EDTA, it has a nitrogen CH2 carboxylate group. That is the main arm of the chelating agent. What we found in this, and you'll get in this story, so you won't have to explain it as we go on probably, is that any chelator that has that structure, EGTA or even the amino acid glutamate, which is 10 millimolar in the brain, will mimic this effect. EDTA just has to be the most potent, and EDTA is added in many of our foodstuffs. I mean, if you drink Mellow Yellow or drink beer, eat certain things, EDTA is considered an essentially safe chelator. In the dementia dialysis, I worry that this may be involved there, in which people mine heavy metals or they put EDTA in the treatment that they give individuals. There's no doubt that mercury chelated with EDTA, which is in a lot of food, considered environmentally safe. And if you would have asked me 2 years ago, is EDTA a good chelating agent, a safe one, I would have said yes. I knew nothing about it. I can tell you that we have studied a large number of enzymes And in a biochemistry laboratory, you always put EDTA in to, yes.
So, for example, for
All right, can you tell where you are now?
Yes, I know where I am. I'm fine. Thank you.
for
Some of
detox,
the slides dropped out. I'll try to get them back in the order where I think they were.
That's okay. I can fake it. Just make them in or so we can.
So I can put them in any order then?
Yeah, just put them in. That'll be fine. Well, otherwise, I don't know how you'll find out. They're not written down in order.
I'm trying to keep them as best as
This is an excellent example of the best laid plans can go awry. I mean, I had that in the zone, but yes.
so for lead detoxification, you wouldn't suggest EDTA?
I would suggest we find others. Yes, other
In
chelators.
the meantime, I mean, right now, if a person comes
There
in
are-
with heavy lead poisoning, would you use EDTA or not?
If I were doing it, if it were me or someone that I had control of,
Yeah.
so like my children, I would definitely want a dithiol-chelating reagent used versus an EDTA one. The dithiols won't mimic this effect, and as a matter of fact, they'll reverse it. Dithiol-chelators.
For example,
I have them back in
Okay.
order as best as I could. I'll
That's-
stand here, and if we need to change it, I'll change it.
This will be fine.
for example, could you give one example of
Yes.
that?
Pardon?
Could you give one example of that?
I will when we get here. We'll talk about some, just about any of them that would bind chelate. I mean, there's a large number of them that are made. I don't have them all listed, and sometimes they call them different names. It's not.
Murky?
I push one button in. Can you push a button back there and make it change? I mean, that might.
When you push the button, the slide locks down.
Uh-huh. Uh-huh. Sure. Sure. Somebody asked a question? Yes.
It's shown recently that EDTA also frees up methyl mercury.
It also what?
It also frees up methyl mercury.
It frees it up?
Yes, sir.
Uh-huh.
Methyl mercury, if you were testing methyl mercury instead of the inorganic mercury, it's three species of inorganic
Yes.
mercury, two species of organic mercury. If you test the organic mercury, which we know is generated down in the mouth, you will find that it binds with these proteins you're talking about.
Mm-hmm.
And therefore it is
Yes. Would you want to get someplace there where there's a microphone? I'm tethered. I can't.
No, I just want to say that, and I'll shut up, okay? It does bind with the SH radical of the proteins. Those proteins you were testing up there, brain proteins, and the enzymatic proteins, it does bind with that to the SH binding, and that's been proven recently by quantum mechanics.
Mm-hmm.
So next time you run your test, if you'll test methyl mercury instead of the inorganic mercury, I think you'll come up with more positive damaging evidence to the brain.
Mm-hmm. Yeah, no, what he's talking about is the reactivity of methyl mercury and even mercury. That's the number one. Yes?
Each day the EDTA cements heavy metals to the brain.
I wouldn't use the word cement. What I think happens is that the three-dimensional structure of mercury chelated to EDTA is mimicking something that is very toxic to tubulin. Now, what this gentleman's talking about is exactly right. Free mercury will react with any enzyme that has a sulfhydryl. When you chelate it with EDTA, conventional wisdom, as a biochemist, says this will no longer react with EDTA. If you're doing enzyme assays, you always throw a little bit of EDTA in to chelate heavy metals to keep them from interacting with the sulfhydryls and inhibiting all enzymes that have sulfhydryls. Conventional wisdom is mercury chelated to EDTA is not reactive. What I'm showing today is that mercury chelated to EDTA is disastrous to tubulin polymerization by the process of preventing GTP from binding to the subunits. And the unique thing about this is it's very selective. That we can totally wipe out tubulin interacting with the nucleotide that's needed to keep it in the right structural form for neural life, for neuronal cells to live, at the same time not affect any other enzyme in the brain that we've tested so far.
methyl mercury will.
Methyl mercury will probably react with everything.
You bet.
But no, I'm not saying it wouldn't. I'm saying it would react exactly like you're saying. I'm saying when we chelate mercury with EDTA, which is a common food additive, that it selectively makes it neurotoxic for that one protein. You won't see any of the other proteins we photolabel change at all, nor is their enzymatic activity changed. But tubulin polymerization is shot, and it selectively does that. Yes?
As a former MS patient, 6 years after mountain removal, 2 years ago, a physician was put me on EDTA chelation, and it made me very ill. And I just decided after 10 treatments to just stop it. And gradually all that disorientation, everything that was brought on by it.
Mm-hmm.
And he felt, well, I know, but he felt that that was going to help me in the long run. But it was making me far too sick, and I didn't have time because of what I've chosen to do. So that sounds to me like I'm, could have become an Alzheimer's victim.
Okay.
Dr. Hennig,
Yes.
we discovered the problem. A few of the slides are cardboard. They fray, rather plastic, and they fray jam down in. Will hand
Okay.
manually operate for you, and one jams will release it.
Okay. When we talk about the tubulin the work we've done of the tubulin that does photolabel, it labeled in 7 of 7 normals. We've done a lot more than that at this time, ALS, PICS, and several other diseases. And we had 7 of them that labeled very lightly. Could I have the next slide? Or do you want me to do it? You do it. Okay, this is one that's out of order, so we're going to jump over here. This is a screen. And what this screen is, we're going to be jumping back and forth. It's not going to be quite in order that it should be. What you can see is we put down here several different metal ions. The first half of it, from here on over, is where we've tested mercury, cadmium, manganese, lead, aluminum, and magnesium. And we added 5 micromolar metal ion. We had no EDTA to this point. And you can see that cadmium was the only one that had any effect on the photolabeling of any band, and it decreased the photolabeling of the beta subunit of tubulin. Now from this part over, from here on over, and this is even mercury, this is 5 micromolar mercury. From this part over, we added EDTA, 100 micromolar, and now in this process you pre-chelate it. You add the mercury to the EDTA, it's 100 micromolar, and we put in 5 micromolar metal ion. The formation constant of this is 22, the log of the formation constant, which would be no free mercury. The minute we did that, nothing else had any effect until we got over to the mercury, and then it decreased it by quite a bit. Can I have the next slide? We'll get around this. Okay, here's the one that should have been in front of that. When we were adding, in this particular one, we had 95 micromolar EDTA added only in the last, or the 4 slides from here on over. The rest of these had either magnesium added, where we have the pluses, I don't know if you can see it from the back, and the mercury concentration. So going from this direction, starting right here, going all the way across up to 100 micromolar mercury, we found out only when we got to 100 micromolar did we have any effect on the tubulin, and it decreased it quite a bit, but it did not wipe it out. And at 56 micromolar, it had almost no effect at all versus the level. Now, if we go over here to where we added EDTA, pre-chelated with EDTA, if we added 1 micromolar mercury, we totally wiped this particular one out. And this was an experiment maybe not done so well, but as we went across, and we'll go back over this again as we get to higher concentrations, we started getting a lot of non-specific labeling. Can I have the next slide?
What was the EDTA concentration?
94 micromolar. We try to stay near 100 micromolar. Just go ahead. This is something I was going to talk about, the neurotoxic effects that have been published with mercury. They don't talk about the effect of it. Most of you will be very much aware of this, but they talk about the blood-brain barrier, etc. But all I was going to talk about that if mercury starts out chelated form, it can be very toxic and can go through this much faster than, say, mercury that's not chemically modified or mercury. Could I have the next slide? This is just telling you that 7 patients with Alzheimer's disease, this should have been way early, didn't photolabel at all. And the reason we started doing this mercury work was that there was mercury in Alzheimer's disease. It was done by Bill Marksbury and Bill Lehman at the University of Kentucky, and they showed that mercury was elevated in certain portions of the brain of people with Alzheimer's disease, that there was a slight elevation of mercury. It was the only ion they could find that was elevated. Can I have the next slide? Okay, here again is another screen in which we're photolabeling. This again is the beta subunit of mercury. Again, we have aluminum, lead, magnesium, manganese, nothing added. And then when we add, these are without, these are with EDTA. When we get over here with mercury, we totally wipe it out. This manganese was a mistake made by the graduate student, because you can see that manganese at 97 micromolar had no effect. And with, again, magnesium, lead, and aluminum at 4.9 or 97, we had no effect. However, the mercury at 4.9 totally abolished the photolabeling of tubulin. Can I have the next slide? This is a comparison of Alzheimer's disease and control brain. And then now we're getting much better with these experiments. We know how to set them up and design them. Again, this is an AD, patient number 1, patient number 2, and we did the same experiment where we increased the mercury from 0 to 6.1 micromolar in the presence of preformed with 100 micromolar EDTA, and there's no change. You don't see much change in the photolabeling of the beta subunit of tubulin at all, which you don't see from the start of the finish. Now, if we go over here to 2 control brains, which do photolabel at 0 concentrations of mercury, even with the EDTA there, if we add 0.8 micromolar, it's decreased about 60%, and by the time we get to 2.3, it's almost totally abolished. So at the same time, we look at the band that contains the glutamine synthetase and the creatine kinase, other enzymes that bind ATP, we see no changes in those nucleotide binding proteins. And it's only in this particular one, and it's a very dramatic change as you're going from several tens of thousands of counts down to nothing with just a little bit of mercury. I would also point out that these are done the same way. They're done at 0 degrees. We preform the mercury EDTA complex. We put it in with the cytosol or the homogenate we're looking at. We wait 2 minutes or 5 minutes, depending on what it is, and then we shine light on it. This is not something that you have to let set up. It almost happens instantaneously, actually. And we'll talk about the time in just a few minutes. But this is a tie-in. You can see that this system right here looks exactly like an AD brain photolabeling. Could I have the next slide? This is a comparison, again, of a control brain, and we're going with the 2 zeros in the center. Again, this is the beta subunit tubulin. This is glutamine synthetase and actin in this region. And as we go across this direction, we are increasing the concentration of mercury in the presence of EDTA, going this direction, pardon me, and with EGTA this direction. And you can see that EDTA is quite a bit more effective than is EDTA in that it blocks the photolabeling quite a bit better. And this highlights the fact that it's very selective for tubulin, has no effect on these proteins. Even if we go up to very, very high concentrations, this stays effectively able to interact with ATP. We've checked the enzymatic activity of glutamine synthetase and actin. They're very happy with mercury chelated to EDTA. It doesn't affect their system. Creatine kinase isn't affected. We haven't found any other enzymes that are affected by the mercury EDTA at this concentration. Tubulin polymerization, though, is shot at very low concentrations. Could I have the next slide? This is a screen of chelators. Now, what I'm looking for, if you've followed my research at all on the Alzheimer's thing, there's 2 things that we have found. Number 1 is that tubulin doesn't photolabel in most AD brains very well at all. It's always decreased. The second thing we found is that glutamine synthetase is extruded in the cerebrospinal fluid, and this is a diagnostic test for Alzheimer's disease now, and it's been given to the University of Kentucky. What glutamine synthetase tells me is that glutamine synthetase is that the brain is suffering from excess glutamate. That's the reason you overproduce glutamine synthetase, to get rid of this neurotoxic amino acid. So our research is designed to try and pull those things together. And what I was curious about, would there be any natural chelators that would mimic EDTA, something that would normally be in the brain that might bind to mercury, methyl mercury, as it got to that position. And here's malate citrate, PDC, we'll talk about these names, EDTA, NTA, EGTA, without any mercury. And you can see that only if we get high concentrations or chelate the magnesium, we start seeing a slight effect with the EDTA. But when we get over here and we look at these things with 2.8 micromolar mercury added, that it's only EGTA and EDTA, and a little bit NTA works, but it's not nearly as good. The other chelators, malate, citrate, PDC, they don't work. These 3 have in common the fact that they have that nitrogen CH2 carboxylate structure similar to an amino acid residue. Now, I'm not showing the data because we're on that, but we also tested the amino acids. And only glutamate and aspartate will give the same effect as EDTA, but they have to be at much higher concentrations, and that's because they're a single wing. They're not chelators, they're just a single wing, but they will actually mimic this effect. Yes?
Another thing here, you're inhibiting both fractions here, though.
On this one, we're chelating too much. We're getting rid of the magnesium. As you'll see later, we have to come back and explain some of
Tubulin and the
This is because we're getting rid of the magnesium with these things. We'll show that later because we're adding magnesium and show how we can reestablish that. Could I have the next slide? This is the formation constants of these things to magnesium, and that's what we're going to explain. You just jumped on my question right away. If we add EDTA, NTA, these other things, we find out that if you have the higher ones there, if they bind the magnesium, they will prevent the photolabeling also of the ATP binding proteins, because they're all magnesium requiring. However, if we compare the binding affinity for the molecules, here's one that doesn't work. Here are two that do work. So it's not the affinity that they bind mercury that causes the thing. It's the structure that's the most important. And we've done several others that aren't on this slide. I just don't have the slides made for that. And so it turns out that we need the like I say, the nitrogen CH2 carboxylic acid group for this to work. Could I have the next slide? The structure here that we're talking about is this part structure here, which can look like part of an amino acid residue. And if we don't have this, we don't get this effect. This caged effect with EDTA is definitely the most potent form of inhibitor of tubulin polymerization. And so we're kind of directing this toward the fact that there are other chelators that don't work that bind mercury just as tight, and perhaps those should be used in a lot of the studies where we're removing heavy metals from toxic materials. Could I have the next slide? On this one, here's the point where we're talking about the photolabeling of the bottom. You've got to understand these probes see exactly what the biological system gives them. If you have here an AD brain, which we pulled out of our freezer, you can see that we photolabel this and we don't label the tubulin. And that's because this is an old brain. It's been around a long time. And as you take a tissue and you freeze it and you store it, the ATP, the GTP, and all the endogenous nucleotides start getting hydrolyzed by enzymes that use them. When you get a fresh brain, you get a system like this where you can photolabel tubulin because GTP is not very high in these cells, but the endogenous ATP level is high, so you don't see photolabeling down here. It's got nothing to do with inhibiting it. It's got to do with the endogenous nucleotide. This was an experiment done on very fresh controlled brains. Not aged brains, not brains that I was standing in the autopsy room when they were dissected and immediately frozen, and we checked them out to say that there wasn't something happening to tubulin in AD brain because somebody wasn't taking care of it, or in the control because somebody, you know, they're taking care of it in AD brain. So as we did this one, again, in these two controls going from 0.62 and 6 micromolar, control 1 and control 2, you can see that we totally wipe out the photolabeling of tubulin even in the freshest brain that has had the best care to show that it's not been abused or anything. Okay, could I have the next slide? If we look at the time or the mercury concentrations that will do this effect, the thing is, mercury doesn't get to 1 micromolar level in your brain for the most part. I don't know what the highest concentrations are, but we're going to go back and say how much do we have to have a disastrous effect. And if you look at this, we don't need to worry about the AD. This is on a gel we have. There's absolutely no photolabeling of this. If we take the control 1 and control 2, we can see that with the concentration curve it comes down very dramatically, and we get total inhibition somewhere between 1 and 2 micromolar, at least the maximum amount we'll see on these gels. Now, so we know the concentration is very dependent. It's in the low micromolar range if we look at it this way. Could I have the next slide? And what we're doing here is the time of incubation. The idea being that we're adding this, and we're doing all these experiments rote, you know. Put in the homogenate. You can either put in the magnesium and the probe and then add the mercury and wait, or and you stop at a certain time. And what we were wondering, would lower concentrations be more effective if we did 10 minutes or 20 minutes? So we took the concentration of mercury that with this particular brain, and by the way, each brain is a little different. Humans aren't like rats. You can't raise them and feed them all the same thing, and they don't all die of the same diseases. And control brains, like I say, they're from people that are ill. So we get a slight difference each time, but not much. So what we did with this, we put EDTA in each one of these samples. We left magnesium out of half of them and put magnesium in half of them because there seems to be an effect of the magnesium, which you saw in the slides. 3 micromolar mercury in this particular case was left out of the first 2, and then but in all the rest of them except the last 2, and we let these go 5 and 60 minutes on the first 2. Starting here, at this particular one here where we had mercury and EDTA and no magnesium, and we put it at 2 minutes, we still have, you know, considerable photolabeling of the of the beta tubulin. When we got to 5 minutes, it was totally abolished and leveled out. So it is a time-dependent factor. So if you left it in, if you're eating mercury, if it's in a live animal, it's there for a considerable period of time. Over here, if we had magnesium present, and we had it in 2 minutes. It still was quite high. As a matter of fact, it jumps up quite a bit. And as we go across, it takes it now 15 minutes to get, you still have considerable labeling. By the time you get to 35, you've abolished it, and at 60, it's totally gone. And you can see that the bottom band doesn't change very much at all at this time. And these two controls on both sides, from 5 and 60 minutes, don't change at all. So we're not getting proteolysis or anything like that. This is definitely effective mercury, and it's very time dependent. Now, we've done this experiment several other times. You can add very, very small amounts of mercury, much less than 3 micromolar. You can go down to fractions of a micromole, and the length of time that it sets with the tubulin, it takes the tubulin out of solution. I mean, it'll take the tubulin from its ability to photolabel with GTP. So the levels we're talking about are much, much higher than you would need for neurotoxic effects. And if you consider that we're preventing tubulin GTP from binding to all the microtubules, and all you have to do is inhibit the polymerization, take out one rail of the railroad track, and it no longer is operational. So I think tubulin is mercury chelated with the EDTA is very lethal to these cells. Can I have the next slide? If we look at the time, and this is adding mercury EDTA after the probe plus and minus magnesium with increasing incubation times, what we see is without magnesium presence, you get an immediate inhibition. And if you have a lot of magnesium and you add the probe first, that it takes it a long time, but it doesn't protect it forever. It just slows down the rate that this occurs. So when we talk about the effect of this material, it's a time and concentration dependent effect, and you can have very, very low concentrations if you want to sit around for days and still get a maximum effect. Could I have the next slide? And it's also slowed down by mercury. As a biochemist, the questions people ask me, what effect, where's it going, and how's it doing it? So what we want to know, does having the probe present or the GTP site occupied slow down the rate of mercury inhibiting or binding to the site? Now, where this would become important, if you have, if you're older and if you have an ischemic attack, the first thing that happens is your ATP levels and your GTP levels drop. And this exposes the sites of certain enzymes to free radicals, things like heavy metals, etc. It makes it much more susceptible to inhibition. That's the reason we're doing these studies. And if we look at this, if we add mercury before the probe without magnesium, we can see that if we did a concentration curve, it goes down very rapidly. By the time we get to about 1.6 micromolar or 3 micromolar, we've totally wiped it out with the length of time of letting it incubate for about 5 minutes, I think, in this case. If we add mercury after the probe without magnesium, it slows it down quite a bit. I mean, it goes a lot slower. However, if we add mercury after the probe and with magnesium at the same time, we get considerable protection with time. Although, as we showed earlier, it still will wipe it out. So what we're worried about here is that people who are lower in energy, if their brain is losing the levels of ATP it should have, they are more susceptible to the toxic effects of mercury. Could I have the next slide? This just shows the data plotted over there. As we can see, without the magnesium and without the probe being added first, we go down quite rapidly. And this one's going down if we let it go long enough. If we let it, say, for instance, in the hour that we showed, it would be down to the same level to where it would give total inhibition. So nothing totally protects this. Could I have the next slide? Again, this is when we added mercury before or after the probe and add the probe last. We can see this is out of context from where we had the thing, that with no magnesium, here we get less labeling to start with, and it drops down very quickly. With EDTA after magnesium or EDTA before magnesium, we can see that we still get a very rapid drop, and this is with the probe being added last. So, and this should have been earlier in the talk somehow. But this just shows how rapidly that will drop if you don't have the probe present. So energy level in the brain cells is very important to protect us against mercury and mercury's toxic effect after it's chelated to EDTA. Can I have the next slide? So the whole take-home point of this story is that in a normal brain, GTP displaces GDP, causing these alpha-beta tubulins to form a microtubule, which is important and essential for neuronal health. In AD, we have clearly shown, and we've studied this a lot, we probably have 100 samples, 100 ADs that we've looked at, and about 80+% of them will not bind, will not photolabel, will not have this exchange occur, and cannot form this structure, or at least a normal aspect of this structure. And if we put in EDTA chelated with mercury, we can mimic this very, very same effect. This does not mean that mercury EDTA causes Alzheimer's disease. I want to express that. But it means it's something that really needs to be studied. And micromolar levels of mercury in any source, as a scientist, it makes me very nervous. I wouldn't care about how I was getting exposed to it. I would not like it, and I don't like having EDTA in my foodstuffs either.
You're saying that this does not show that EDTA mercury causes Alzheimer's disease. It doesn't show that it doesn't either.
It doesn't show that it doesn't, no.
So, I mean, it could possibly.
I think it could cause a dementia.
But not an Alzheimer's dementia.
Well, it might be an Alzheimer's dementia. I'm not that smart. I mean, I'm not that well informed. I want to be very careful about what I say. I think it's an area, as I say, that needs studied. It is definitely not a healthy situation. If you have a material in your body that can inhibit tubulin polymerization in neuronal cells at submicromolar concentrations that seems not to be protected by high concentrations of GTP or magnesium, is time dependent. You know, it may explain what we mean when we say somebody's mad as a hatter. If they're exposed to mercury, sooner or later your neuronal cells will start dying. Cells that don't have such a dependency upon tubulin, and brain cells are probably the most dependent cells on tubulin, they work quite well for a long time. But your neuronal cells are very sensitive to this system, to this toxin. And I would classify mercury EDTA as a very potent neurotoxin, biochemically. And from what I've read, people say mercury EDTA goes through the gut, gets into the bloodstream, passes the blood-brain barrier faster than, say, inorganic mercury. As a biochemist, that makes sense. The body has a lot of sulfhydryls, and it may be that in the proteins, and they bind mercury very, very tightly if it's free. However, if you chelate it, it can't be scavenged by these free sulfhydryls that may be in things like albumin and other molecules. When you chelate it, you allow it to exist long enough to get through these protein barriers that we build up to protect the brain, where it can get into the neuronal cells and cause some severe damage. I think there are a lot of things that cause Alzheimer's disease. I don't think it's all mercury EDTA. I mean, it would all be something such as mercury necessarily. But it definitely, I think this data shows that the conventional wisdom, mercury chelated by EDTA is non-reactive and non-toxic, is not true. I mean, absolutely. I absolutely believe that. And I think it can cause a lot of disease. It probably doesn't do a lot of the other cells any good either. Could I have the next slide? Oh, here. Well, now you'll see my real weakness. I'm a very good chemist. I'm a lousy cell animal researcher. But when we found this data, we went hunting around saying we're going to start feeding rats mercury. Everybody wants an animal model. Ned Kasarkis at the University of Kentucky was doing some research on Lou Gehrig's disease, or amyotrophic lateral sclerosis. And he had some rats that he'd fed mercury to, and he took them to the very end until they were about to die, and whacked off their heads and body parts and did some stuff, but he froze the brains. And so we got the brains from Ed's rats. They weren't designed, and they weren't fed EDTA either, unless it's in their chow that they feed them. These were just put mercury in the water. And so we looked at those, and what we can see here are the two control rats. Here's one here, he labels very well tubulin. This control, by the way, doesn't label so hot with tubulin. But when we got the HG fed at 9 months and 10 months, there's no photo labeling of the tubulin. And the same thing over here. Here's a couple other HG feds. This is nothing like a normal rat brain. Most control rat brains look like this. They have an extra added protein up there that humans don't have, which makes them rats, I guess. But we were very excited about this, so we did our own study. And I did this with a medical doctor friend, and neither one of us are very good at animal research. And we, also we didn't have a lot of money, and I just thought, well, you feed them mercury and check them out. But what happened, could I show the next slide, and we'll explain these as we go along. Here's what their protein profile looks like. You can see there's not any difference, and please notice the one on the outside here. He's very healthy. He was killed at 4 months, and the reason we killed him at 4 months is because the graduate student and the MD, and I wasn't involved in this part, I was just going to do the biochemistry, is that whenever they started looking a little sick, why we thought, well, we're going to lose him, so let's kill him. Don't let him, we don't want to go and find him dead. So we didn't, we shot ourselves in the foot, essentially. So if you look at this guy, he got sick, and we cut his head off at 4 months, and the rest of them went the entire sick term. We killed them at, we killed some at 8 and 12 also. I'm not going to talk about that, but I have the first slide. But just remember, this is the guy that got very ill, and this may be part of what Alzheimer's about. And you can see there's no, EDTA or mercury EDTA present. Pardon me. Am I? Oh, there's. This isn't the slide. This has got the wrong thing on. I'm sorry. Anyway, this is where we photolabel all these animals. We don't have the EDTA present. This shouldn't be 70 micromolar. This is the photoprobe. And you can see this is the only one that didn't photolabel. Is the one that got ill and died. So at the bottom, I got the wrong slide out of this. I mean, but anyway, this guy here did not photolabel and die. This is the months of treatment. This is what we fed them. They had 70 micromolar mercury EDTA in their drinking water. I'm still going back to the biochemistry for a period of time. This is the controls that did not have it but had EDTA. So the 6 that had EDTA were all very normal. Of the ones that had mercury EDTA in their drinking water, 70 micromolar, we could find nothing wrong with them. You can see some of them are up and down, except the one that got sick and we had to kill early. And we killed him at 4 months, and he was very ill. At the 6 months time, though, we started taking out the animals. Like I say, we made a mistake. We did this 6 months, 8 months, and 12 months. Always the ones with mercury EDTA had lower tubulin photolabeling, but not enough that you'd want to say anything about it. But when we went into the batch, instead of setting the rats off in cages and saying this one's going to be killed at 6 months, this is going to be killed at 8, and these are going to be killed at 10 and 12, we kept them all in one big cage, and we took the ones that looked the sickest and killed them first. I didn't do this. I mean, and so I think we didn't do the experiment right. However, it may also be that there may be something else that has to happen to you. It may be that there are certain, maybe 10 of us could suffer an exposure to mercury, and only 1 of us would get Alzheimer's disease or get a neurotoxic effect. Some of us are very strong to certain toxins. A lot of us can drink a lot of alcohol. Some of us can't drink much at all. And I think this rat may represent that one situation, maybe a fortuitous infection of some sort, or some other thing that I can't explain. Probably a lot of people in the audience could explain this better than me. So we're repeating this experiment, and we're going to be much more professional about it, getting some help from people that know some animal toxicology studies, and look at this. So this is but this is the one effect we did have. Earlier on, all of them that were treated until they were very ill, none of them photolabeled. Yes?
When will you use animals fed low levels of selenium and zinc then?
I'm sorry, I'm not.
Will you use animals fed low levels of selenium and zinc then? Because that
you know, I will tell you the truth. I won't have anything to do with this. I'll do the testing. I'm going to find somebody that I can do collaboration with that knows all of the. I know what you're talking about. The selenium would be a very helpful thing, if indeed this would prevent the toxicity. But I'm not going to do any more animal studies. I'm going to help and collaborate with somebody that does these, that knows what they're doing.
If you're working with a person who's going to be working with you, would they send frozen brains to you or what?
Yes. Frozen brains are fine. You know, just take them out and get them stored as quickly as possible. That's basically what we need. So I think the animal studies are wide open, and I would encourage anybody here. Boyd Haley's not going to, I stepped out of the bounds of my competence here, and it shows dramatically. It's embarrassing to put this work on the thing, but I was going to tell you that there is something here, and I think it should be studied, but by someone that knows what they're doing. Okay. Could I have the next slide? I think we're through. Yeah, this is some of the examples of stuff that have EDTA in it, and this is my graduate student slide, and I think it's very prominent, and I think it's something that we should work at, and I think it's something that people should be made aware of. Chelated mercury with EDTA. EDTA's in our foodstuffs. It's sprayed on the vegetables you buy, and it does do a good thing. It does chelate cadmium and certain other heavy metals, lead, etc. And so I'm not against chelators, believe me, but I think that we need to look into this, and that people who are working with the dithiochelators have the best approach. I would point out that if we inhibit a brain, the tubulin, with mercury EDTA, that we can reverse that with dithiochelators. Bring it almost back to normal. We have also taken Alzheimer's disease brain. Now you got to understand, these people have died, and they've been exposed this a long time. We can recover about 20% of what would be considered control levels of photolabeling of tubulin by putting a chelator in with the Alzheimer's disease brain. We cannot get anything near 40 or 50% back, though. It just doesn't happen. So that tubulin, after being, say, precipitated or out of normal for a while, something has happened to it, probably by intracellular enzymes that makes it non-functional. But you can have some recovery of this, of the binding. Could I have the next slide? I think we're going to have a blank here, and we're going to